All right, let me get started. So today I'd like to first of all thank the organizers, thank you Martin and everybody for organizing this wonderful workshop. Today I would like to talk about some very recent work in my lab that was triggered partly by two of my students' interest, Bo and Jason, who really wanted to work on single cells, and also by the availability of some very exciting new technologies, in particular the 10x gene code system that can be used for producing large-scale single-cell RNA sequencing data. And we are in a collaboration with 10x, so we started looking at this type of data. So, as we all know, DNA sequencing and RNA sequencing has been around for a very long time. Traditionally, DNA and RNA sequencing is done in bulk populations of cells, which typically involve thousands to many more cells. And DNA or RNA is extracted and then sequenced by sequencing technology like Illumina. More recently, single-cell techniques have been developed which attempt to solve the challenges of obtaining some material from single cells. And those involve, first of all, isolating the cells and then amplifying the DNA or RNA to get enough material for sequencing, and of course, barcoding. There is many different systems that have been developed literally over the past year or two for single-cell RNA analysis. Those typically involve some kind of microfluidic technique to isolate the cell and mix it somehow with reagents or beads so that amplification and sequencing can be performed. I want to make a parenthesis now and talk a bit about the 10x system for those of you who may be unfamiliar. 10x originally was developed for DNA sequencing, and it produces data of some very powerful form that I want to just spend a minute to explain. So 10x microfluidically mixes beads with DNA material coming in very long, in molecules of a range of lengths, but they can be very long, of average length, 150 or 200 kb. And so those are placed in water compartments immersed in oil. So here you see an actual photo where you see the beads, those circles here, and a few compartments actually don't have a bead, very few compartments, only 10%. There is typically 1 million compartments, each one with a unique barcode. Amplification, barcoding is done within each compartment separately. Then emulsion is broken, and the material is ready for sequencing. What is cool about this is what you get is read clouds, what I like to call, or linked reads. Basically, you get clusters of reads that contain all the same barcode because they came from the same compartment. And each compartment only covers a tiny fraction of the target genome in very long molecules. So essentially, you get the following thing. You get what I like to call read clouds. So if you align those barcoded fragments to the target genome, they come in clusters or read clouds that are very, very far apart from each other. So you know which reads came from the same long molecule. And this is very powerful because now you can do phasing, structural variant detection, detection of variants within repeat regions of the genome and segmental duplications, because you can align a whole barcode uniquely to a copy of the sectu, and so on. I want to emphasize something because you may be also familiar with synthetic long reads. There was a protocol with which we worked also, the molecular protocol developed at Stanford, which produces synthetic long reads. So basically, reads are barcoded like that in compartments, and from each barcode you get a very high coverage of reads per long molecule. And then those reads are assembled because they are in such high coverage, so you get synthetic long molecules, long reads. The problem with this approach, DSLR approach, is that you require large coverage of the genome with long molecules and a large coverage of the long molecules with reads. So if you then do sequencing with Illumina, it will be 50 times more expensive than doing traditional Illumina. Another way to do things, and which I believe is much better, is to obtain read clouds. So you get very sparse representations of each long molecule, and then you want to pool the read clouds with some computation, with some algorithms to obtain the same long-range information but at much lower cost. Okay, back to single cells. So the 10x protocol can be used for doing single-cell RNA sequencing, though this is a product that was released very recently, I think like a couple of months ago. And the way this happens is basically beads are mixed with single cells and again in water droplets emulsed in oil, and cell lysis starts immediately as soon as they are encapsulated. The microfluidics have been worked out to work quite well, so basically most of the compartments have beads and the cells are loaded in a Poisson way. Each cell has a unique barcode and each gene has a unique molecular identifier from which basically with that you can tell which, you can basically get reads that tell you which genes are present in the cell. This is extremely high throughput. So tens of thousands of cells are processed in a single run. The caveat is that compared to other approaches, such as in-drop and drop-seq and so on, this is much more sparse data. So it's a powerful type of data. Extremely high throughput, like 1 to 2 orders of magnitude higher than the previous methods, but also much more sparse. We are talking about 95% sparsity versus 20 to 60% sparsity. So how do we use single-cell RNA-seq data? What is the goal? Well, the goal is that when we do bulk sequencing or bulk RNA sequencing, we really are averaging over a potentially very high heterogeneity across a cell population in a tissue. For example, a cancer tissue could have normal cells and many different subclones of cancer cells. Or a blood sample has T cell, B cells, monocyte, different types of T cells, killer cells, and so on and so forth. A disease tissue may have lots of healthy cells and some disease cells. So when you do comparison between normal and disease tissue, you want to be able to tell across all these different types of cells. Also, cells can be in different stages, such as different stages of the cell cycle. So it's really interesting to look at the heterogeneity in a cell tissue through RNA, single-cell RNA sequencing analysis. The challenges are also, they are of two kinds. They are the technical challenges, which is great sparsity, very noisy data compared to bulk, and also challenges having to do with the amazing, there are so many different ways in which we may be probing cell heterogeneity. So in disease versus normal settings, in tumor settings, in different types of tissues. So the data looks extremely different in every different experiment and in every different technology. So what do we do? One approach is to do supervised analysis. So basically, we go to ground truth generated by, say, ENCODE, roadmap, and so on, and we compare the gene expression of each of the cells to known gene expression patterns across different tissues. Perhaps by having some marked genes, annotated genes that we know are characteristic to be in a particular state in different tissues. However, this misses potentially new discoveries in cell heterogeneity. So really, perhaps it is more desirable to do an unsupervised analysis to discover in an unbiased way the biology within a tissue. So to do that, it is very important to get the cell-to-cell similarity measure right. So how do we measure a similarity across two cells of the expression pattern? Problem is, if you use something like Euclidean distance or Pearson correlation, some standard metric, it may produce vastly different performance depending on how much missing data there is, how much dropout, whether the amplification bias gets some genes to show ridiculously high expressions, and so on. Huge outliers and everything. So it is not clear how to do this, how to develop a method that works well, a fixed method that works well in every type of data set. So what we did is we developed a method we call similar, which basically learns, and it stands for something, I forgot just now the acronym. It stands for, it basically learns a similarity measure aimed to cluster cells well. So basically we look to find the similarity measure across the cells that makes the matrix of cell-to-cell similarity be of block structure. We don't have any priors, and then that similarity measure can be used readily, if it is already in block structure, for clustering, dimensional reduction, and visualization. So let me take you through the method, really briefly. So this is an overview of similar. We start with a matrix of n cells by m genes. Think of n and m being like a few thousand, a few tens of thousands. And we learn a weighted combination of multiple distance metrics or kernels. So basically we will learn a weight vector to weight between different distance metrics. And we throw here, as you will see, a kitchen sink of distance metrics. We throw 54 different distance metrics, so as to learn a block diagonal structure of a matrix. So we also learn the block diagonal structure at the same time as we learn the weights. And then we can use this for clustering and visualization. Okay, so here, this is the objective that we are optimizing. And I will walk you through this. So we basically, one thing to keep in mind is we have a problem of minimizing over the similarity S across every pair of cells, and that's a symmetric positive matrix of similarity. And each cell distributes its similarity across all the cells. So basically the sum of similarities across all cells, from a cell to all cells, is 1. Okay. We also learn at the same time a weight W, which weighs between all the different distance metrics, and I'm going to explain precisely which is our collection of distance metrics. So we learn the weight W, and we also learn a low dimension matrix L. L is of dimension C. C is an input parameter by the user, which specifies how many clusters you want to find. And in our experiments, it looks like our performance is quite robust to C, but think of C as something between 5 and 20, something small. So we learn this matrix L as well, which is a low-dimensional matrix, and that's where L participates. So let's go through this objective function first of all, to see what's going on. So basically, the cell-to-cell similarity matrix is based on multiple distance metrics that are weighted, and we learn this set of weights. And basically what's happening here, because this is a minimization, you want to make the similarity high whenever the distance is low. Okay. This is simply a regularization parameter on S, and this enforces the rank constraints. So basically, this is the symmetric normalized Laplacian of a graph where nodes are the cells and edges are the similarity values. So the higher weight the edge, the higher the similarity. So this is the symmetric normalized Laplacian. And by taking the trace of this expression, you make L want to cluster the cells into C compartments, where in each compartment C, the similarity is very high, so as to minimize the trace across, because when the similarity is high and this is negative, you minimize the trace. Yes?
Can you explain what is Sij versus Dxixj? Like you seem to have both distances and
Yes,
similarity.
yes. So basically, imagine that you have the similarity to be fixed now between the cells, right? If the similarity is fixed, what you are doing here is you are trying to minimize the distance across the most similar cells. Imagine now that the distance is fixed, and you are learning S. Well, now you want to learn such an S that maximizes the similarity across the least distance cells.
But
So now,
I guess my question is, why do you need S if you are going to learn the weights for the dxij? Why do you even need this S?
to be able to basically to be able to have this expression where we
And you
enforce.
couldn't do it directly on the D?
Well, yeah, we can talk about it offline, but this worked well for us. So this is a tri-convex problem. So we are alternating between optimizing for S, for L, and for W, and we circle through these three until convergence. In practice, this works in the very, very fast. So we have no problem to analyze thousands of cells in tens of thousands of genes, such as in 10x data.
How do you enforce the normalization on the S's? They're like probabilities, right? Sij.
Yeah.
So how do you enforce that when you do the optimization?
This is basically this. It works. So we can take it offline. Basically, if you keep everything constant and you do that, then you can basically enforce. Yes.
Can you explain what L is?
L is a low dimensional matrix. So basically, L transpose L is the identity with C entries. And so basically, L in every one of the components, and there is very few, imagine 10 components, will pick high values in a cluster of cells. So L picks high values in a cluster of cells that are very similar, and the more similar they are, the more negative the trace will be for those. So it will enforce them to be a cluster. Okay. So let's keep going. So this is our distance metric. So we have 54 different kernels. Each one is a Gaussian kernel with two hyperparameters. One is the number k, which is basically you are comparing a given cell with the k nearest neighbors of the cell. And the other hyperparameter is essentially the variance. So we have three levels of standard deviation, 0.5, 1, and 2, and we have how many? So basically, we have like about 20. We have 18 different values for the nearest neighbors k. Okay. So how well does this do in practice? You have 11 cell population. We basically tested against 4 data sets for which ground truth is available in recent literature. And one of them is on 11 cell populations of neural cells and blood cells. That's 249 cells. Each of, you see, there is different experimental protocols in each of these data sets. Another one is neuronal cells with different sensory subtypes, embryonic stem cells with different cell cycle stages in this case, and pluripotent cells under different environmental conditions. So we have a variety of different tissues and experimental settings. So one thing I want to point is that when we learn the distance metric, we do not really converge on a specific kernel. So here, for the first data set of 11 cell populations of neural and blood cells, we basically learn a distance metric that is a composition of many different values of k, and to a lesser extent, many different values for the standard deviation. So we really seem to be learning to be adjusting the the distance measure quite a bit in this case.
What's
This
K?
K is the number of nearest neighbors for which basically in this
Okay.
yeah, K is the number of nearest neighbors from which you take the variance. Okay, so this is the block structure if you take the highest value for the kernel of the highest value. This is the block structure if you take a low value kernel. And this is the block structure we learn by our combined distance metric. So this is the matrix similarity, the similarity matrix S. So we looked how we do compared to Euclidean and Pearson correlation. We basically computed all pairwise similarities and we sorted cells by the ground truth. So here are the cells sorted in X and Y axis by the ground truth. As you see, we get very clear block structures in every one of the data sets, while Euclidean and Pearson do not get very good block structures. And then we compared clustering performance in comparison to several different methods, varied methods, convex and non-convex clustering methods, subdimensional reduction methods. And for each one of those dimensional reduction methods, we clustered using k-means, where k, in this case, was the ground truth. I want to point out that performance of similar did not vary very much if we give C to be substantially different from the ground truth k. So we did several experiments, I'm not going to show them. But basically, similar seems to be quite robust in the choice C. And if anything, you want to overestimate C rather than underestimate it. Okay, so how do we do? We measure performance in two different ways. One is the normalized mutual information between the true labels and the cluster labels. And the other one, so basically the highest value of mutual information, the better the technique does. And the second way is the nearest neighbor error, basically how closely the label of the cell is similar to the label of the ground truth label of surrounding cells.
So how do you know the right answer? How do you know
Great
the
question.
ground truth?
The four data sets that I am describing here all have ground truth, either because of separated populations of cells or because of fact sorting and so on. So in each of the four cases, we know the ground truth.
But even if somebody, sorted something on facts based on some markers, it might still be that the whole gene expression pattern from some guy that was
Absolutely.
faxed in one bin is closer to some guy who was faxed in another bin.
Absolutely. Very good question. So there could be errors in the ground truth. But to the extent that we uncover the ground truth in an unbiased way, because we don't have any supervised learning here, so we cannot be learning any artifacts because we are getting clusters, and then we see how our clusters have
Sure.
similar
Okay.
values according to the ground truth. So we couldn't possibly be biased in learning something strange about the ground truth. So, but yeah, that's a very good question. How do you know ground truth in single cells? And now you have to go to the technical details of each of the experimental and labeling protocols. So how do we? So basically, these two normalized mutual information between our unsupervised clusters and the ground truth labels cannot possibly be biased. Also, nearest neighbor error, or how close the assigned ground truth label is of each cell to the surrounding cells according to our similarity, again, that cannot possibly be biased by some artifact of the ground truth. So in a way, if this clustering performance is high, it also shows that the ground truth labels were reasonably good. So how well do we do? So this is the normalized mutual information, and this is the nearest neighbor error. And as we see, in almost all cases, similar does substantially better than all of the other methods, and in particular, if you notice, it does substantially, or I would say dramatically better than the second best here. So this is 95%, versus the second best being 77%, and so on. So we were quite happy that we could match ground truth in these 4 datasets. We can also use similar for dimensional reduction and clustering and visualization, and methods that have been used traditionally for this application is, of course, PCA that everybody is familiar with, and also another very good method, TSNE, which is basically a nonlinear projection method. Let me go into a little bit more detail in case, for those of you who are unfamiliar. So TSNE, or T-stochastic neighbor embedding, tries to minimize the KL divergence between the actual data, where basically the data is turned into a probability distribution as such, versus a low-dimensional projection of the data. So basically think of the YIs are low-dimensional projections of the original data. What we did is we took TSNE, and to apply our method in a very similar manner, we simply hacked TSNE, you can modify it very easily, to replace this similarity measure with our similarity measure. So now our defined similarity measure comes directly from our learned similarity matrix. So how do we do? What I'm showing here is again the 4 datasets with ground truth being colored, and here I'm showing similar on top, TSNE, PCA, and ZIFA, which is another method that was developed recently, published in Genome Biology a couple of months ago, by actually the first author being Emma Pearson, who is also a co-author of our similar method. And as you see visually, it looks like similar in each case, in each of the 4 datasets, does better than the other methods in producing a dimensional reduction that leads to a nice intuitive clustering and visualization of the data. We also did another experiment comparing with TSNE, where we created a simulated dataset of basically these 4 clusters, and then we performed similar in TSNE without noise or dropout, with noise and dropout. Dropout is basically when you lose a lot of entries of the matrix. And as you see, it looks like similar does better than TSNE, is more robust to noise and dropouts. Okay, and then finally we looked at the high throughput 10x data, where we had 4,500 peripheral blood mononuclear cells produced by Chromium TM system from 10x Genomics. There is a high level of sparsity here. What you see here is the count of UMIs, unique molecular identifiers. This is the count, this is the number of entries that have this count. As you see, 0, a count of 0 is the dominant here. So basically 96.9% of this matrix is zeros. Only 2.1% is a ones, and then small values for higher values. We compare here, it's really tough to do a comparison because we really don't have ground truth here. So we use as a proxy of ground truth the comparison of every cell with DMAP, which basically is different blood cell types, and we look at the expression profiles. These are the labels produced by correlation with DMAP. This is the labels produced by similar. We see that we get roughly similar clusters. This is projection done with TSNE, not with similar. So we want to do we want to basically we want to have a picture here where similar does not take part in the projection. So as we see how it does compared to our proxy of ground truth. We found major cell types such as T cells CD4, CD8, B cells, monocytes, and progenitor cells. Further, when we look at clustering each of the cell types, so we took the monocytes and we clustered them using C equals 2, 3, and 4 further with similar, and we get 2 clear clusters. It's not clear to us what they mean biologically, but that's what we got. We did the same analysis with B cells, 2, 3, or 4 clusters, and we got 3 clusters in this case. In T cell CD4, we got 4 clusters that seem to be reasonably separable. And T cell CD8, we got 3 clusters. And then there were a number of cells that could not be annotated by DMAP, and they seem to be outlier populations. So that's all I wanted to say. So now in the works, together with 10X, we are looking to establish some ground truth for blood and basically perform some ground truth experiments to see further how well this method does in clustering high-dimensional data with lots of dropout. That's all I wanted to say. I want to thank my students, Bo and Jason, and our collaborator Emma, who developed this method. Also our collaborators in 10X. And again, this is a very new, it's the first time I give this talk, and we are really, really excited. If you have ideas of single-cell data that you may have that we could use and collaborate, or if you just want our software to try it yourself, I would be delighted to talk further. So thank you, and I'm happy to take questions.
Thank you.
Bill?
So this is very interesting. I think, to me, it seems like one of the main ideas here is you're putting a prior on your projection that the data exists in clusters. Right?
Yes.
So I guess the question is, in what context is that a reasonable assumption, right? I mean, a cell cycle is not necessarily clustered, for example. Cancer is pretty heterogeneous, and there might be continuous, distributions among clonal populations, whereas peripheral blood, maybe there really are distinct cell types.
That's a very good question, and I would probably answer it over the next several months, mostly empirically. Cell cycle, I think, is reasonably clusterable, I would imagine, because different stages have clear, distinct patterns, but there may be some smoothness across the stages. So the question is, how well would this method perform? Where would the cells in the boundary be? It's not precisely, I would answer this empirically, and it would be interesting. So yes, we do assume that cells come in discrete clusters rather than being some smooth transition across the clusters, and that's an important assumption of this method. Yep.
So I was trying to understand exactly this S matrix. It's a little mysterious, right? So if you think about k-means clustering, there's like the raw data, then there's your similarity metric, like D, and then there's the clustering, right? And so you added an extra layer in between the distance metric and the actual clusters, right?
Yeah.
Is that partly to deal with, say, outliers, like things in the data that would throw off the similarity measure? Because, that matrix by itself is, because that will be heavily biased by the clustering that you impose on it, right? So how do you interpret this S matrix? Is it a technical thing, or is it
it's very, very good question. Is the S matrix the clustering, or is it something that can be used for clustering?
Or something in between?
Yeah, essentially the S matrix is more or less clustering the data, although after you get the S matrix, you could imagine, for example, I don't want to flip to too many slides now, but as you saw in some of our data sets, you get a block structure that has sub-block structures. So I wouldn't actually call the S matrix the clustering. You could then use whatever criteria you want of how many classes you want to get what you call a cluster to further subdivide. And you can even apply this approach as we did in the last data set hierarchically. So take a cluster and then further subdivide it. So we are still exploring. We're very new in exploring this method. This is all the results, all the plots I show we produced over the past month. So there's more analysis to that. Yep.
It's very interesting. I just missed something about the optimization of W. I don't know. Can you go back to the equation just to? Sorry.
Yes. Let me see.
The optimization.
Let's see. Let's go quickly. Yes.
Yeah. So you say you optimize in S, then in L, then in W, and in
Yes.
direct, right? But in W, it seems that the objective function seems to be just a linear function of W.
Yeah.
And the constraint is that it's set to 1.
Yeah.
So in this case, I think the optimum should be to select just one distance. So the optimum of a linear function over the
Oh, the W you mean?
W, yeah.
W.
So I don't see how you get selection of many. You showed later that W is spread across different distances, right? It seems that just the optimization of this function in W should lead just to selection of one distance. So is there something not written here, or something else, or expression of why it doesn't pick just one distance?
But no, this. So basically, let's take it offline. Because, yeah. Yes.
So out of all the kitchen and sink distances, do you find some of them more useful than the others? You have
Yeah, good question.
distance
Again,
measurements?
that's an empirical question. So here, in this particular dataset, I wish I had the same thing for all datasets. The most useful, the highest weight is for K to be 24. Now, intuitively, what does this mean? What is K equals 24? It's basically saying that when you look at a given cell, you look at roughly 24 neighbors to take its variance in expression. Okay, what does this mean? Why would we want to be varying this across different experiments? We want to be varying this because if you have very high sparsity or very high noise, then maybe you want to be looking somehow at, if you have very high noise, you want to be looking at more neighbors if you have low sparsity. But if you have very high sparsity, but whenever something is close to you, you really trust it, then you're going to go lower in K. So that's the thing, that different experiments, depending on their dropout and their level of noise, have intuitively they would pick different local neighborhoods to inform the clustering of the data. Yes?
So how big is the impact of cell cycle? So we have been doing similar analysis. We would try to take out the impact of cell cycle first, and then.
So this is one.
clustering.
This experiment is done on the cell cycle. So this basically is cells that are in 3 different cell cycle stages, sorted by facts for markers of the cell cycle. And I don't know the impact of the cell cycle in the other datasets, but I can. But this dataset is a cell. Oh, wait a second. This is the Böttner dataset, so that's the cell cycle dataset. So we got 3 pretty clean clusters here. And in terms of accuracy of the cell cycle, we are at 89% neighbor mutual information and very, very low error rate, 0.05%. So only 5% of the cells have neighbors that are not of the same cell cycle. Yeah. So.
In the comparison to Euclidean and Pearson, how do you account for the fact that you have many more parameters to optimize that?
Yeah, I mean that is not necessarily a fair comparison. It's just something to show that our similarity metric gives you block structure, while that block structure is not immediately obvious by some Euclidean distance or Poisson correlation. So we just wanted to make a simple point that if you take a naive similarity measure, you don't see as clear a block structure.
Is there some way that you can now go back and see what mixture of metrics you chosen, and to maybe make a simplified, reduced version of this algorithm where you now basically use it to find out and say, aha, these are the key things that this is selecting?
Yeah, we did. So we did that. So basically, when we started this method, where is the, here. So when we started this method, our kitchen sink here included a whole bunch of Pearson and Euclidean and different things. So we threw more, and then we realized that all the other kernels were not needed. We were converging on a combination of Gaussian kernels. I don't quite know why this is the case, but it seems that those Gaussian kernels seem to be doing well, but within those, we seem to really need multiple hyperparameters. So that's an empirical question of why this is the case.
Because in the end, let's say if your biologist that produced the data comes to you and says, why on my data set is this the right way of clustering? What does it tell us about the biology?
You could actually interpret things, because the standard deviation values, the distribution of standard deviations, let's say that they are centered more on a given value, it means that it tells you something about the actual standard deviation, I would think, that is appropriate for this data. And the different values of K will tell you something about the local neighborhood sparsity of the data. how many cells is informative to look around to call. So this basically tells you when you compare two cells, their expression value, you want to normalize by some kind, you want to basically take into account how much two similar cells vary. And to get that measure of how much two similar cells vary, you look at the local neighborhood of k similar cells. So I think it's somewhat interpretable in your data to some extent, but yeah, it's very hard for me to give you a concrete answer.
Okay, maybe we should move on. Thanks, Sarah.